Thank you very much for inviting me. It's a great pleasure being here and talking to you, to the audience who is most motivated to hear what we are doing. So my name is Tatjana, and actually shorter version is Tanya. I come from Institute of Cancer and Genomic Sciences at the University of Birmingham. So let me just introduce our team first, which works on various aspects of biology of chronic lymphocytic leukemia. I personally work on DNA repair aspect of chronic lymphocytic leukemia, and I will talk in details about what it actually means. And my particular focus is in finding way to translate our research into a clinical practice. So everything that we're doing has an aim to end up in clinical trials. We have a great support of basic scientists such as Professor Grant Stewart, who made several discoveries in DNA repair field of new chromosome instability syndromes, new predispositions to tumor development, and the same with Professor Malcolm Taylor. So they're really basic scientists. You met this morning Professor Paul Moss, who is a great support, great collaborator, our chief CLL physician, who provides us over the years with the CLL samples advice. And our second important collaborator is Dr. Guy Pratt. You might have met him as well. So I'm not going to give you a classic introduction in chronic lymphocytic leukemia. I just want to point out two things. One is that chronic lymphocytic leukemia is characterized by aberrant accumulation of mature B lymphocytes, as you all know. But what is important, what I want to point out, is that these lymphocytes, these pathological tumor lymphocytes, circulate between blood, peripheral blood, and between lymphoid organs and bone marrow. The difference between cells in peripheral blood is that they are quite idle. They don't proliferate. They just hang around. They don't die very easily, as you probably know, but they just maintain the bulk of tumor mass, but they don't proliferate. They're not in charge of disease progression. The population of CLL cells that is in charge of disease progressions are those that are in lymphoid organs. Because unlike many other tumors, CLL cells require a lot of support for their survival and proliferation. You probably know that as well. The second important thing that I want to point out is enormous clinical heterogeneity. And of all of you present here today, your story may be similar, but they're also very different in terms of speed of progression of disease. Some of you might be diagnosed with chronic lymphocytic leukemia and just being watched over time without ever requiring any treatment. Some of you might be diagnosed and requiring treatment after a period of time, but disease progression is very slow, and we call this indolent chronic lymphocytic leukemia. The unfortunate people, however, not majority, where disease is very progressive. Very quickly there is a requirement for treatment, and treatments tend to be not very successful over time. But as I say, this is not majority of people. And certainly this picture is likely to change with the new available treatments. However, it is important to note, and this is what we observed, one of our clinical fellows last year, that there is a group of patients with complete spontaneous regression, less than 2%. But people presented with a full-blown chronic lymphocytic leukemia, which over 10-20 years become spontaneously cured. So we are very busy trying to understand what is actually happening in this group of leukemia, because this is how people should be really treated. What the body invents itself as a natural cure. Right, you will understand that we as a scientist want to know what is really behind each of these trends. And as I said, chronic lymphocytic leukemia cells interact with their surrounding in lymph nodes and in bone marrow where disease proliferates, where disease progresses. This happens through a lot of different interactions. I will not go through all of them, that is not necessary, but it is important to know that participants in this support are different type of lymphocytes that we call T cells, some very large cells from stroma of lymph nodes and bone marrow, which we call nurse-like cells, and fibroblasts and so on. And there is a lot of crosstalk between these cells and CLL cells all the time. And in addition to that, there is a very important factor of stimulation by some sort of antigen. So try to think that in some ways CLL behaves like a chronic infection, where there is a chronic presence of particular antigen, foreign protein that stimulates CLL proliferation. And again, this is happening via a very special interaction. We call it BCR signaling. And this is where your now favorite drug, ibrutinib, works, because it blocks this important interaction. You probably know all about that. So the strengths of these interactions between T cells, nurse-like cells, fibroblasts, and some others will influence how quickly disease progresses. The same goes for a degree of BCR signaling, a level of activity. But in addition to all that, CLL cells themselves undergo a lot of genetic changes, and these genetic changes can also influence disease course. So what are these genetic changes? Please don't be frightened by this slide because I will make it very simple to you. So this is the number of genes that can be mutated, and we know are now mutated in chronic lymphocytic leukemia. Not in all of patients, some of patients, yes, in others not. They can be combined in different way. But when you actually summarize what they're doing, these genes, in terms of function, they regulate DNA response. You know what is DNA? This is source of our genomic material. This is where mutation happen. DNA contains a huge number of genes. And these genes have to be translated into proteins that do the function, that maintain cellular survival, proliferation, and so on. So all these mutations happen in the genes that either conduct the message from DNA for protein synthesis, or, also very important, if DNA is damaged, which happens all the time, during normal metabolism, during normal function of the cells, then some of these genes will make sure that there is either repair or cellular death. So these are the mechanisms around the DNA and its regulation that are core to the biology of genetics of chronic lymphocytic leukemia. So our own focus of our research is on two particular genes called ATM, that is short for ataxia telangiectasia mutated, and comes from the name of the syndrome called ataxia telangiectasia, that is characterized by progressive neurodegeneration and 300-fold increase in development of tumors, leukemias and lymphomas. In early 90s, I was involved in the cloning of this gene. We did clone it, And since then we studied its function in details. And around 2000, year 2000, we were able to show that this gene is not only related to the syndrome where its mutations are inherited, people are born with both copies inactive, but also in general population, such as yourself. Gene can be mutated later in life in tumor cells. So what does this gene or protein do? When DNA is broken, and as I say, this happens all the time during our metabolic processes, or if you receive chemotherapy. Chemotherapy works by the way of breaking DNA. ATM recognizes that ends are broken, and this protein now interacts with a number of other proteins, making very important decision for the cell, which is either to repair broken end. For that, the cell has to stop proliferating, and if damage is too large and cannot be repaired, ATM makes sure that the next protein is activated, called P53. You all know about P53 and very nasty mutations. And this P53 now makes sure that a cell undergoes cellular death. So if there are mutations in either ATM or P53 gene, damaged cells will survive. So if you receive chemotherapy, you do not have this mechanism of activating cellular death, and this leads to poor therapeutic response. So what we know now, these are your two chromosomes. ATM chromosome lies ATM gene lies on chromosome 11, and P53 on chromosome 17. So these are cytogenetic abnormalities that your doctor is looking for, particularly if there is a disease progression. They want to see whether patients have deletion in chromosome 11 or deletion in chromosome 17, because these abnormalities are an indicator that function of either ATM or P53 is not normal. You also see here that in addition to 11q abnormality, you need to have mutation on the other allele, and only then you completely lose ATM function. And that's not the case with P53. You can have only one mutation and a defect might occur. So to prove this scientific hypothesis, we looked at the patients on a largest CLL trial that ran some time ago, called CLL4, UK CLL4 trial. And patients on this trial received different type of chemotherapy, either chlorambucil or fludarabine cyclophosphamide. They were divided into different arms. So we analyzed over 300 different patients for this study, and we looked at the mutations in ATM and P53. And what you can see clearly here, that if both chromosomes are affected for ATM gene or P53, we have a very poor survival in patients. So that was clinical indication that our hypothesis and our knowledge of P53 and ATM function is right. So what is actually happening if you have complete loss of ATM or P53 function? You have such a loss in a smaller number of cells at the beginning of disease. They're presented here in red dots. And you give such a patient chemotherapy, you kill all other tumor cells that don't have ATM or P53 loss. But these cells over time start to taking over because they haven't responded. So although you have at the time of chemotherapy this illusion that you shrunk tumor load, actually over time you didn't, because nasty ATM and p53 mutant cells take over. We call this clonal selection. And doctors became aware of that. And I'm sure you heard this morning for Professor Moss that particularly for p53 mutant tumors, we have other solutions. So let me just very briefly remind you what you heard this morning, that we went a long journey from classic chemotherapy towards more sophisticated chemotherapy, towards inclusion of immunotherapy, to these small molecule inhibitors that many of you might be receiving nowadays. And that is a great story. For a vast majority of patients, management is hugely improved. But we mustn't forget a category of unlucky individuals that are still suffering from relapses under these treatments. And this is the case with p53 in particular, and to some extent with ATM, depending on the study. But this is one with ibrutinib resistance, suggesting that both events might be important. So what I'm actually trying to do here is to make understanding that we still need novel therapeutic approaches. So, what is this? London Underground. I'm very proud. You recognize it immediately. So this is how I'm going to try and explain you our therapeutic approach. So if you imagine that is between DNA damage and survival, which are on the opposite end of London, there is a network that you can use to arrive. However, you would appreciate that there is a direct road and there is an indirect road, right? And everyone prefers direct road, how it should be. So if you imagine that responses to DNA damage governed by ATM and p53 to survival are direct road, then if this road is blocked, we need to use other ways for cells. Cell needs to use other ways of survival. And we were very busy to try to understand what are these other networks in the CLL cells that has ATM or p53 mutations that are developed as a compensatory mechanism, if you like, dependency mechanism. Because our hypothesis is if we inhibit now these other roads, then there is only one option, which is complete jam of the traffic and cellular death. So this is how we think. And I will just give you two examples to make a point. So the first strategy, some probably of you heard about PARP inhibitor olaparib, because it is used in other solid tumors, particularly of breast, of ovarian origin, with a great deal of success. Let me just very briefly explain what's happening. So PARP is a protein that deals with a single break of DNA. And if we block PARP by olaparib, these breaks now turn to be more nasty, become double-strand breaks. This now requires ATM and p53 function. Normal cell will be able to do that because this mechanism is functional. But cell defective in ATM will not. And hypothesis is that will lead to cellular death. ATM is not the only protein that interacts with PARP. There are some others, but for this story today, ATM is important for chronic lymphocytic leukemia. So we've done a lot of preclinical studies, and I will show you some of animal experiments. I know that some of you are very reluctant to sign for animal experiments, and I sympathize with animals a lot, but they give us a huge amount of information, because what we can do, we can actually inject human leukemia or human lymphoma into animals. So one of you can be recapitulated in animal. We can try different treatments and see what happens if we treat such an ATM null tumor with olaparib, completely disappears. So this was in preclinical studies. We were very encouraged by this. And this coincided with the availability of a very good inhibitor which can be taken as a tablet or capsule at the time. So we started a trial, and Dr. Guy Pratt was in charge of this trial, which was supposed to be phase 1, phase 2 study, and you know that phase 1 study is really exploring toxicity of the drug, and phase 2 study is exploring activity of the drug in a very specific context. So we included here not only chronic lymphocytic leukemia, but few other relatives that have a high frequency of ATM loss, mantle cell lymphoma and chronic prolymphocytic leukemia. And as I said, we had two goals, mostly to determine whether drug is safe enough to use in patients with leukemia. I don't know whether you've ever seen the layout of phase 1 trial, but it's basically recruitment of 3 by 3 patients going through different doses up to the point when it's not safe to give it to the patients. If there is any single toxicity, there is a need to repeat recruitment. If toxicity happens again, it goes back to the reduction of the dose. So this is how trial works until we determine what dose is right. So we were able to complete phase 1 of this trial, and that was published in British Journal of Hematology. I have to tell you that recruitment for just toxicity phase was really very, very ill people that exceeded all other options. And even within that very ill cohort, we were able to see that if patients had loss of ATM function, they were staying longer on the trial, and some of them had quite decent responses. Unfortunately, it didn't last long enough to make substantial effect, but the trend was very encouraging. We haven't conducted stage 2, which would be the most informative of this trial, because then we run into competition with the magic drug, ibrutinib, and so on and so forth. But this stays an option, and perhaps in combination with some very low dose chemotherapy for patients who don't have any other options. So that is one story. The second story is inhibition of another pathway that is of another protein called ATR, ataxia-telangiectasia related. It has a similar role to ATM. I will not go to both of them. It interacts also with p53. In other words, if ATM and p53 is lost, then ATR protein is a principal in response to replication errors. And replication errors are DNA errors that occur during replication, during synthesis of DNA. So when we inhibit ATR pathway, we have the same effect. Cells die. So we conducted preclinical stage of this trial, and here you see some animal experiments showing very nicely that we have very good killing for ATM and defective and p53 defective tumors. And if you directly visualize what is happening in the cells when we add this ATR inhibitor, this is your DNA synthesis. This is visualization of your DNA synthesis, very powerful methodology. You see how it starts all going very badly compared to the cell where we don't give inhibitor, and cells starting to die in a very nasty manner, which we call mitotic catastrophe. So I just want to finish by saying that we demonstrated in this study published in Blood selective killing of ATM and p53 deficient tumors, and what is very important that we demonstrated in this paper that there is a synergism with ibrutinib, that this drug potentiates effect of ibrutinib and the other way around. And consequently, there is an impending trial Professor Hillman will initiate with a special arm for ATM and p53 deficient CLL tumor, where a combination of ibrutinib and ATR inhibitor will be given. So we have these new options for a very poor prognostic leukemia. So in summary, what I showed you, that inactivation of principal damage response genes is frequent, causes chemo resistance and shorter progression-free survival. We identified so far that inhibition of two proteins may help in killing these subclones, this subpopulation of CLL cells. We conducted a phase 1 clinical trial with PARP inhibitor, and we are about to start trial with ATR inhibitor. So thank you very much. There are a lot of collaborators here. This is my group, and this is where I used to work, and we used to clone ATM gene, but now I work in a slightly different field. Thank you very much.